Okay, hello everyone, I'm Shiela. Today I'm going to talk about habitats and fish communities at mesophotic depths in the Mexican Pacific. This research was published in Journal of Biogeography in 2020. Before introducing the research content, I want to let you know what mesophotic zone is. It's a region of low light from approximately 30 to 150 meters beneath the ocean surface, just like the red bars in this image. Why do we need to know about mesophotic zone? The importance of mesophotic zone can be summarized in these two reasons below. It can serve as a potential ecological refuge from overexploitation or climate change, and represent a unique ecosystem worthy of focused management. Due to the limitations in research funding and technology, our understanding of mesophotic zone is limited in terms of technology. The mesophotic zone is relatively shallow compared to the depths explored by large deep-sea submersibles. We can see in the right image that the California Current and the North Equatorial Current flow along the western side of Mexico. The complex hydrographic mixing of water from the California Current and North Equatorial Current, along with deep water races to sunlit depths due to the steep seafloor and underwater canyons, are the two reasons that allow tropical and temperate species to coexist nearby. There are two survey sites in this study. The first is the Bay of La Paz, also called BLP. And this survey focused on the biogeographical provinces of Cortez. It means the biota exhibit similarities in taxonomic classification and evolutionary history. And the area indicated by D and E in the figure represent BLP. And the second survey site is the Revelatojo National Park, also called RMP. And this archipelago consists of 4 islands, and this research focused on 3 of them, which labels A, B, and C in the figure. Both BLP and RMPs are regions known for high marine biodiversity. And there are 2 purposes in this study. The first is to investigate the diversity of fish species in the biogeographical provinces of BLP and RMP. And the second is to compare the influence of reef habitats and non-reef habitats on fish diversity patterns at mesophotic depths. They use a portable, remotely operated vehicle in this research, equipped with lights, cameras, and sensors that measure temperature and depth. At each site, they descend RV to the seafloor and begin roving swim surveys, which is highly effective in quantifying species diversity. Next, I will play a short video showing the operation of the RV underwater. This footage comes from the manufacturer of the RV used in this research.
Because people always talk about how little we know about our ocean, but they tend to ignore the reason why. Those who are curious enough to ask big questions haven't always had the tools to start answering them.
And the crew on the board can remotely control the RV using an operating panel. And here I will briefly compare 2 underwater survey methods in terms of maximum reachable depth. The RV used in this study could reach depths up to 100 meters. And this refers specifically to the RV used in this research. Other RVs may reach different depths depending on their specifications. Untrained scuba divers can typically reach a maximum depth of 30 meters. The investigation time for RV depends on battery duration, whereas scuba divers are limited by scuba tank capacity and diver stamina. Using RV for underwater surveys can allow for the observation of smaller and faster moving species. And as shown in the right picture, Each dot represents one study site. Most of the sites are located around these 5 islands. The sites were chosen based on the sea surface, clarity, and bathymetry. They compile information from Robson and Allen, Ocean Biogeographic Information System, and fish base to identify fish. And the algae was identifying using herbarium records. The numbers on the pie chart shows the number of species found in each family in this study. Serenity has the highest number of observed species with a total of 10. Other families such as Carenidae, Labridae, Lutjonidae, and Pomacanthidae each had 5 species observed. In total, this research recorded 66 fish species across 81 transects. This research also found a new geographical record of the Galapagos snake eel. It was previously only registered in the Galapagos and Cocos Islands. And the Galapagos snake eels was observed twice on video during the surveys around Calarian Island. And the following is a short video showing actual RV operation conducted by this research team. And this one is the Galapagos. And this figure shows depth observations of fish species. The red line represents the maximum depth reachable by scuba divers, which is approximately 30 meters. In other words, species found below this line are less likely to be recorded through diving surveys. And the horizontal lines means the average depth for each species. And the boxes represent the 25th and 75th quartile values, meaning 50% of the observations for that species fall within this depth range. And the 2 vertical lines above and below the box represent the standard error, and the points represent outlier values. Most fish species were found throughout the survey depths. However, some fish species prefer shallower depths, such as surgeonfish and pufferfish. And some fish species prefer deeper depths, such as snapper, grouper, and other related species. And in this picture, richness refers to the number of fish species present in the area. And this image shows fish species richness by temperature and habitat type, including reef and non-reef habitats. And points represent transects, while lines represent a linear least squares regression of richness by habitat, which is a statistical method. And the shaded area indicates 95% confidence interval. When the shaded area is narrower, the model's predictions are more stable, making the result more reliable. The results indicate that richness increased with temperature and was greater in reef habitats than non-reef habitats. Let's review the purpose of this study. The first is to investigate the diversity of fish species in the biogeographical provinces of BLP and RNP. And this research identified 66 fish species in total and extended the known depth range of 9 species. And second is to compare the influence of reef habitats and non-reef habitats on fish diversity patterns at mesophotic depths. And this research showed that fish species richness was significantly higher in reef habitats than in non-reef habitats. And here I'd like to talk about why should we care about fish diversity. First, fish diversity helps increase fishery stability. Studies have shown that fisheries targeting a wide range of species tend to have more stable catches compared to those focused on a single species. Second, fish diversity holds significant medical value. Compounds derived from marine organisms may play a key role in future drug development. And third, changes in fish biodiversity can alter the structure of marine ecosystems. Recent research suggests that such changes affect ecosystem dynamics, including the biomass of species at lower trophic levels. The main conclusion of this research is that expanding the use of ROV technology can enhance monitoring efforts, ensuring appropriate habitat-specific management and conservation. For instance, some areas may need to be designed as protected zone, while others could be open for fishing. And this is my presentation. Thank you for your listening.
I think you do a very good job, and the presentation was smooth, and you made a good use of the mouse to guide us to know the figure. But I think the sound, the voice was a little bit low. I cannot clearly hear what you say, and that's all. But you did a good job. Thank you.
Okay, thank you for your presentation. And this content is easy to follow, and you add many figure with animation and make it really colorful, make it really interesting. But your talking speed is a little bit fast, so I can really follow it. And in some figure, you mark some of the position, but the color is really dark, and I can really see it. And that's all. Thank you.
And I think your design and your content is great, but just yes, you need to slow your talk speed and you need to make it louder because during your talk I need to concentrate more what you said. Yes, and also your tone needs to be more energetic. Yes, thank you.